excited to have you here. We look forward to your presentation. I'll stop my share right now and hand it over to our first presenter. Thank you so much.
Hey, good morning. Can you hear me? Okay. So thank you, Kathleen, and the colleagues for the presentation and for the invitation to be here today. It's really nice to talk about beta diversity and biodiversity for you guys. So I'm very excited. Let me start my presentation. Okay. Okay. So today, I'm gonna talk about beta diversity and its components in freshwaters. The time is short, but I will try to talk about scales of diversity, beta diversity definition, beta diversity metrics such as Whittaker, Sorensen, and Jacquard, beta diversity partitioning in components replacement and nestedness, Permanova and Permdisp, and the local contribution and species contribution to beta diversity. And then we will practice these different topics in the R program. Biodiversity can be measured in different scales, such as alpha diversity, as the number of species in a site, gamma diversity, as the number of species in a region, and beta diversity, measuring species variation across sites or time. For example, take these two lakes. The circles are individuals, and each color represents a different species. So what are the alpha, gamma, and beta values? First, what is the alpha diversity of each lake? Alpha diversity is the number of species in a single place. So the richness in each lake is 3 species. The green, red, and yellow in the first lake, and the red, purple, and yellow in the second lake. And about the gamma diversity, we have the species red, here, yellow, green, and purple. So the gamma diversity, considering these two lakes, is 4 species. And beta diversity. There are many metrics to calculate beta diversity. One of the first metrics was proposed by Whittaker. Here, beta diversity can be measured as the ratio between regional and local species diversity, and is calculated by dividing gamma diversity by the mean alpha diversity. So, for example, what would be the value of beta diversity in this case? Think about it. Well, the gamma diversity is 4 species, and the mean alpha diversity is 3 species. So, beta diversity is 1.333. If the two communities are completely similar, the value should be 1. And higher the value, higher the beta diversity. This is the oldest metric of beta diversity. But today, we have many ways to measure beta diversity. Some of the most commonly used beta diversity metrics are Sorensen and Jacquard for presence and absence data, and Breikert's for abundance data. For these metrics, 0 is a total dissimilarity, while 1 is a total similarity. So let's see this index. The letter A here represents the number of shared species between the communities. The letter B is a number of species unique in site 1, and the letter C is a number of species unique in site 2. So here, what is the number of shared species between these two lakes? How many species we have in common between these two lakes? You can type in the chat if you want. So we have the red and the red and the yellow species present in both lakes. So the number of shared species is 2. Now, what is the number of species unique to the first lake? Well, the number of species unique to the first lake is only 1, because it's the green species, only the green species. Finally, the number of species unique to the second lake is also 1. Only the purple species is unique to the second lake. Now we can calculate the similarity of sovereignty according to this index. Here is the index. Now we know what the A, B, and C means. It's very simple. And we can calculate it. So 2 multiplied by 2 is 4. Wait. And 2 multiplied by 2 plus 1 is 6, according to this index. Finally, 4 divided by 6 equals 0.666 of similarity, or 66% of similarity. The dissimilarity is the complement. So if we have 0.666 of similarity, we have 0.333 of dissimilarity. So Jacquard would be very similar to Sorensen, but is not multiplied by 2, right? Here in the index. But it's the same, we use the same arguments to calculate the Jacquard similarity. Well, Jacquard and Sorensen are one of the most common examples of dissimilarity metrics, a way to measure beta diversity. But today we have many metrics. And to choose which metric use will depend on the type of your data, if it's presence and absence or abundance. You can also try to control the influence of difference in species richness between communities using turnover component or other metrics, but there are many options of dissimilarity metrics. Also, beta diversity metrics such as Sorensen and Jacquard can be partitioned into 2 components, turnover and nestedness. We have different approaches of beta diversity partitioning, such as Baselga and also Podani and Schimera and Cardoso approach. Turnover measures the replacement of species among sites. Here, the green and the yellow species are replaced by purple and pink species. So this is the turnover component. Nestedness measures if one site is a subset of another site, or measures the gain or loss of species across sites. For example, the green species was lost from the first to the second lake. However, we did not see a replacement of species, only a loss of species because the green one was lost. So both components can influence beta diversity, and we can try to understand the mechanisms that drive beta diversity, turnover or nestedness. Also, we can calculate beta diversity within or among treatments. For example, using PermaNova, we can investigate if the composition of a treatment is different from another. Here, the species composition of forest streams, here in purple, is different from the species composition of impacted streams, here in green. This is indicated by the polygons that are distant, so the species composition is probably different. Below, using parameters, we can investigate if the species variability measured as the mean distance of each site to the centroid, here's the centroid, is different among treatments. We can try to investigate the species variability. Here, the variability is lower among impacted streams than among forestry streams, which is suggested by the smaller polygon here for the here for the impacted streams. The lower beta diversity here indicate a process of biotic homogenization. Biotic homogenization process is common in many freshwater ecosystems due to many causes, such as land use, exotic species, or damming, for example. We can also calculate the local contribution to beta diversity proposed by Legendre and De Caceres. Local contribution to beta diversity, LCBD, indicate the degree of ecological uniqueness or singularity of each sampling site. Sites with higher LCBD are more distant to the centroid and may indicate sites with higher priority for conservation, for example. So they are more different than others. Here is a site with lower contribution to beta diversity because the distance to the centroid is smaller. Finally, using Legendre and De Caceres approach, it's also possible to estimate the species contribution to beta diversity, the SCBD. Species contribution to beta diversity, SCBD, represents the degree of the relative importance of individual species to beta diversity across sites and can be used to identify which species contribute the most to dissimilarity among communities or require some specific attention. So let's practice these concepts and approaches in the R program. I will finish here and we'll change. Let me see you. Sharing. Okay. Do you have any questions, or maybe we can just go to the R and then we can ask if you have any question, as you prefer. Is okay so far?
Yes, I think we can
Okay.
move to R. Thank you.
Okay.
Mhm.
Just open. Just here. Here. Okay. Share screen. Share this one. So guys, are you seeing my R?
Yes.
Yes. Okay. Um, if you can open also your R program, then we can try to do together and run together the script. In the beginning of the script, there are the packages required for this practice. Did everyone manage to install these R packages? I hope so. So once the packages are installed, we have to run and load the packages. And vegan, beta part, add spatial, bat, ggplot2, and the ape. It's still loading. They are still loading. Okay. Okay. So the first thing is create the data sets to use in our practice. We will create two data sets. The first one is very simple. We have only two sites, site 1, site 2. Here we are just combining the two rows. And here is just to give names for the species. And finally here we have the data sites. It is the same data set that we used during my presentation that we calculated Jacquard similarity together. It's the same one. So we have two sites, and for species is a presence and absence data set. The second data set is bigger, and we are going to use to calculate it Permanova and Permigis. So here I name it data strings for the example of forcing packet strings. So we can create the strings. So here we just created the 10 strings, and now we created the category. So the first 5 strings are forest, and the last 5 strings are impacted. Now you are just combining the rows to make a data frame. And here in column names, we are giving names to the columns. The columns are the species, and so finally we have our data set. So our data set, we have 10 strings and six species. Is also a data set of presence and absence data. So these strings we call here in category as forest strings, like here, and these strings would be the impacted strings. So far, so good. Here we just created the data sets that we are going to use for the next steps. Okay, so the next step is to calculate sourcing and Jacquard dissimilarity using the data sets. There are many functions in R to calculate the dissimilarity. We are going to use the vectors from vegan package. So here we can see the data. And here is to check the function, how the function works in the help. So we have to provide the argument of the community data matrix, which is data size, and choose a method, which is the dissimilarity index. So here in this line, we are going to calculate the sourcing dissimilarity. But as we can see in the help, we don't have the sourcing metric. To calculate the source dissimilarity, we are going to say to choose the breakers method, which is break here in the method argument. And we are going to say that binary is true. If you are using presence and absence data and saying that binary is true, we are calculating the sourcing because sourcing is the presence and absence version of breakers. So breakers and sourcing are very similar, but breakers is the version for abundance and sourcing is the version for presence and absence. So we can say method is break, binary is true, and we're going to calculate it, the sourcing. Because sourcing is for abundance, which you don't have here. VegDish measures the dissimilarity. So if you remember from the presentation, when we calculate together the Jacquard similarity and dissimilarity, we found a similarity of 0.66 and then a dissimilarity of 0.333. So we did write our calculations during the presentation, and is the same here. We obtain it from the VegDis function. It's very, very simple. And in the next, we can choose other beta diversity metrics, for example, Jacquard. And here we just have to choose in the method the Jacquard option. And now have the Jacquard calculation is a little different because the index is a little different between Jacquard and sourcing. It's okay so far? Okay. You can feel free to ask anything. I am not seeing this chat right now, but we can stop me and ask. Okay. The next step is to calculate the beta diversity components, turnover and nestedness. We are using the data sites again here. And one of the functions to calculate beta diversity components is the beta pair from the beta part package. And this is the Baselga approach. So here's the function. Here you can say the details. And this argument is to choose the family. So here in this line, this row, we're going to calculate the components based on Sorensen dissimilarity. So for that, in the index family, I choose Sorensen. And let's run to see what happens. Okay. So we run, and now we have, I will open it. We have three components of a list. And the last one, beta SOR, is the total dissimilarity based on Sorensen. So it's the same value we found before using our calculation in the representation or using VEGGs. So this is the Sorensen. This one, beta SNE, is the nestedness component. And the first one, beta sync, which means Simpson, this one is the Simpson index, which is a turnover component. So here we can see that our beta diversity is complete due to turnover component and not nestedness. So there are a replacement of species across sites and not a loss of species. And we can do the same thing using now Jacquard component, Jacquard metric, but the name of the components are a little different. Let's run. So here we have the this one is the Jacquard component, the total beta diversity. Beta JTU is the turnover component, and beta J and E is the nestedness component. So also here we can see that our beta diversity is completely due to turnover component, the first one. I put a tip for you guys here. So these both metrics was using the beta pair function, which is the Baselga approach. But we also have another approach which became to be more commonly used nowadays, which is the Podani and Carvalho-Cardoso approach. This approach, we have to use the BET package and the beta function. Here we can see some details in the help. And to calculate it based on Jacquard dissimilarity, we first use the first argument, which is the community. We can say that abundance is false because we have binary data with only present and absent. And here, should say that we would like to calculate the components. So let's see the output of the function. So it's a little different, but here we also have the components. This one is the total beta diversity, which is Jacquard dissimilarity. Here we call replacement component, this one. Also completely due to the replacement component. And here, instead of nestedness, we call as richness difference component, the last one. Is also a 0, as we found for Basel approach. Guys, so far so good. Any question? Sorry, because if I'm going too fast, because our time is short, and there are many things that I would like to show you, but we can also ask him and for Bislo. Okay, so I move. And the next analysis related to beta diversity that I like to use is Permanova. And now we're going to use another data set, which is the data strings, where we have, let me show you all the data, the entire data set here. So in data streams, we have 10 rows and 6 columns. The rows are strings and the columns are the species. Okay. So to calculate it per MANOVA, we can use this function from vegan package, Adonis2. For this function, to calculate it per MANOVA, we need a category, which we already created in the beginning of the scripted. And the category is the type of the string, if the string is forested or impacted. So one way to calculate per MANOVA is using the function, and then we use as the response variable, which is our data set. And finally, we say the factor, which is the type of the strings, forested or impacted. So just to remember, here we're going to see if the species composition from forested strings is different from impacted strings. This is the question. And here, the output, and we can see here in the type of strings was significant. Here we have the F value, R square, your degree of freedom. And this suggests that the species composition from forest strings is different from the impacted strings. Using benthic invertebrates, for example, is very common to see the species composition is different. So per MANOVA to see if the species composition is different. And the next analysis is PermDisp, also from vegan package, and the function is the beta disper, this one. Here, first, we have to calculate the dissimilarity. You can choose any metric of dissimilarity, including the components like turnover or nested components. It will depend on your question. Here, I used again the Jacquard dissimilarity when I said the Jacquard methods. So it's the same we did before, but the function is here. And now we have to say in the first, let's see the function in help. Multivariated homogeneity of groups dispersion is the beta disper function from Anderson. So the D argument is the distance matrix, and the group is the factor variable. So here, this object is our distance. This, I name it this. And here we have the type of string. In our case, we'd like to see if the species variability is lower among the impacted strings than among the forest strings. So let's run. Okay, sorry, I have to run first the this. Just remember this object category is forest, 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 impacted, impacted. So the first 5 strings are forest and the last are impacted. Oops, sorry, I have to run here. And now we can see the output. And here we have to check the average distance to centroid. So here we have the average distance to centroid. And forest strings, we found a mean distance to centroid of 0.32, and very similar to this impacted strings, which is also 0.328. 328. So we can see these results are very similar. So probably and the variability is similar among impacted or among forest strings. To check the significance, we can calculate it. The test, a permutation test. And here we see there is no difference between species variability. So, for example, if I have a question regarding biotic homogenization, because I could think that impact strings have a lower beta diversity, suggesting a process of biotic homogenization, I would not corroborate my hypothesis because they are similar. And here we have an ugly plot. It's very simple to do. Just use the plot and then the object with the beta disper output. But as you can see, this plot's not very beautiful. But we can see here is a PCOA. So permidis in Permanova, the mean distance to centroid and the distance of the centroids. And they are calculated in a multivariate space, which usually is the PCOA. So here the first X, here the second X. And we can see that forest strings and impacted strings are distant, which indicates a different composition as we see in Permanova, but the beta disper function show that the variability, which is the size of the polygons, are similar between forest and impacted strings. So I like to perform plots, graphs, and figures in R. And I brought for you guys an example of a PCOA plot to understand the permidisp, the beta disper, and the Adonis function to better visualize our data. So because this is too long, I will not provide you the details about this code because it's just to perform a more beautiful figure, okay? So you can select all the part of the graph is in the ggplot package, the construction. We don't have time to say regarding each part of the graph, but you can feel free to use or to ask later regarding some specific parts of the code. So here we have another figure, which I think is more beautiful. I think you agree, I believe you agree with me. So here we have the polygons. Green are the forest strings and red are the impacted strings. So you can see they are separate, which indicates a different composition. For example, for benthic invertebrates here, we can have chironomids in the impacted strings, and in forest only stoneflies, for example, so the species composition is different. But the variability is not significantly different. Is the size of the polygon are similar, so you don't have a lower beta diversity in impacted strings. So you don't have a homogenization process. And I'm almost in the end, so to finish my presentation, I also brought for you guys an example of LCBD and SCBD. So we can use these functions from the add spatial package from Legendre. Here are the functions. The second LCBD comp is to compute LCBD from any distance matrix. And the first, I think, is only using Hellinger transformation to calculate species contribution. So here we can use the beta div function. We are using again the data strings dataset. The method is Hellinger transformation, and here the number of permutations. I'm sorry, this comment is in Portuguese, but I'm saying that the beta div for calculation of SCBD only works with Hellinger using this function. So let's see our output. Here we have, oh, sorry. So here we have the local contribution of each site to beta diversity. So when higher the local contribution, more unique is this site. So here we have the contribution of each string. We can try to understand why, for example, this string 1, is more unique. Maybe the environmental, this string is also more unique in terms of environmental conditions, or is more isolated from the others. There are options. And the last part, sorry. Is the SCBD. And so the SCBD is the species contribution to beta diversity. So each value indicates the contribution of each species to beta diversity. So guys, this is, let me see you here. No. So this is my, which I prepare for you regarding beta diversity. There are many other metrics, but because it's only a workshop and the time is short, this is what I prepare for you. But I'm open for questions, and also you can have my email and write to me. I'm completely available. So thank you. I'm free for questions if you have.
Thank you so much for the presentation, Danny. That was, I think, very clear and very, very helpful to walk us through all these scripts. If there are no questions right now, I don't see any in the chat, but we'll wait a couple minutes. Then we can move on to the second presentation.
Yes, I will.
I have a question, sorry.
Okay, there we
Sure.
go. Perfect.
Hi.
Hi.
Thank you very much. It was really useful. And I wanted to know if you have any suggestions about how to choose if using Sorensen or Jacquard, because I totally don't know.
Okay. I think it's hard to say. What I usually do is if I'm going to compare abundance and uh, presence absence data, I try to use the correspondent like Jacquard, Sorensen, Burkhardt, for example. But to choose between Jacquard and Sorensen, uh, I also have a difficult. One of them usually is said to, how can I say, give more importance to rare species. I think it's Sorensen, if I'm not mistaken. So I think Sorensen, you have more importance to rare species if this is something we'd like to do. So I think Sorensen is a better option. But I also I usually try more than one metric and try to show more than one. And usually the output is not so different between Jacquard and Sorensen. But I think one of them give more importance to rare species. I think Sorensen.
Thank you.
But thank you for your question.
Any other questions to Danielle? Maybe later. So thank you. So I think if there are no further questions, we can move on to the next presenter today, Francieli. Thank you so much, Danielle, for that very clear presentation. And maybe there will be more questions later on.
Hello, guys. Can you hear me? Yep. Just a quick comment about the last question, if I can, Dani. It's actually the contrary. So Sorens gives like plus is true, so gives more importance for the common species, and Jacquard it's like an equal balance between common and rare species. So you can choose with that if you want to weight more one or the other.
Thank you for I went to record it when during the question. Thank you.
No problem. I'm very happy to be here today. I would like to thank Leon for the invitation. And today I'm going to talk a little about functional diversity. Just a moment, I will share my screen. It's okay? Everyone can see it?
Yes, looks great.
Okay. So functional diversity is a very comprehensive topic, but I try to summarize here for today for us to talk a little about some concepts, the basic concepts, and how we can choose traits, because traits are the first step that we need to do the selection, to run the indexes of functional diversity. So it's just like a small talk about these main topics, about these main concepts. And if you will have any question, you can ask me during the presentation, no problem at all. Oops, sorry. So functional diversity has been studied for a long time. Since Darwin, for example, he used to classify species by their trophic guilds, but no one called functional diversity at that time. So the past 20 years, some researchers start to define functional diversity, to define what is a functional trait. and to give us some, they propose some indexes to calculate functional diversity. So in this last 20 years, we can observe that had a big, huge increase in studies with functional diversity. So why do we study functional diversity? Because it's a straight way to relate how species contribute or respond to environmental filtering, biotic sorting, dispersion limitation, and how these responses influences the ecosystem functioning. So we can choose some traits for each species, some characteristics, and link these species to the ecosystem functioning. So in a community that we have a lot of species, we have also a range of traits, of characteristics. And these characteristics, these traits, they are, as I'm going to show you, they can be divided by traits that we call effect or response traits. So this composition in the community, after passing to environmental filtering, biotic sorting, and dispersion limitation, we have a convergence of traits or a divergence of traits. So when we have high functional divergence, for example, it means that we have a high degree of niche differentiation. So these species, they are not competing and they are using the resource more efficiently. So this it's linked to an increase in ecosystem functions. So when we have more traits and more divergence of traits, we have more ecosystem functions. So before we start to calculate functional diversity, before we think about functional diversity, I divided this presentation in some steps that we need. So my talk here today, it's more practical. It's for us to know how to choose the indexes and what each index means, how we can use different to respond our questions, our studies, what we are investigating depending on our question. So the first step is defining a set of traits because all indexes are based on these traits. So we need to be very wise when we are choosing the traits because it's not like a recipe that we can just use the traits that are in the literature, or because I read a paper that shows that traits and I'm going to use in my study. No, because you need to choose the traits that will respond to your question. If you are investigating climate change, global warming, which traits do the organism that you are studying will respond or cause an effect in that ecosystem function that you are studying, regarding to, for example, as I said, warming, or if you are studying, for example, an impact of construction of a dam or a building or something like this. So the traits will be different depending on your question, what you are investigating. But what are traits? How can we define our traits? It's important to know the definition for you to choose your traits when you are gathering them. So functional traits can be morphological, biochemical, physiological, structural, phenological, or behavioral characteristics of the species that are relevant to their response, as I said. So if you are investigating some impact, some how that trait will respond to some environmental change, or they can be an effect, how they cause an effect on that environment, yeah, and on ecosystem properties. But which trait should we use? As I said, there are a lot of, depending on the organism that you are studying, if it's fish, for example, if it's zooplankton, phytoplankton, there are so many traits in the literature, so many options that you can choose. Most of them are related to habitat use, reproduction, feeding yields, how they escape predation, how they swim. That can be a behavioral trait. Yeah. So we need to choose very wisely, as I said, because if you choose a composition of traits to run your indexes, and you have, for example, saying that you have low functional diversity, but maybe it's not the picture of the real picture of the nature and the environment that you are studying. It's only because you didn't choose well your traits. Yeah? So arbitrary choices can lead to erroneous conclusions. Well, there is no right and wrong. As I said, it depends on your question. It depends on your goals. So you can have a lot of options and you need to think each one of them. They're the ones that respond right to your question. There isn't also a right number of traits. We are going to see that we need to have more species than traits in a community, in a sample, to run the indexes. But there isn't a right number, if you like 5, 6, or 10. But we need to keep in mind that as much traits as we have, we are also going to increase the probability of having functional redundancy. Because some traits can be correlated. For example, if you are studying the effects of warming in an experiment, and you are going to use, for example, filtration rates, or you're going to use respiration rates, these traits are correlated to body size, for example. So if you use body size and the other two, you need to choose between them and see if they are not correlated. Then if you choose all of them to put in your indexes, you can have functional redundancy. And continuous are better than categorical. So if you have, for example, body size that is a number, it's better if you have the measurement than if you classify like a small bodied or medium and large. It's better to use the real measurement. We have lots of types of traits, so we can we need to know these ones because we are going to calculate our functional diversity based on distances that we are going to create. So to choose the distance to calculate our indexes, we need to know the nature of our traits. So if they are quantitative or qualitative, or if you have a mix of traits, for example. As examples of quantitative traits, we have continuous, such as body size, or discrete, such as the number of antennae or spines, for example. For qualitative traits, we have binary, for example, presence or absence of some structure in the body or some behavior, something like that. For categorical, we could have, for example, the feeding guilds, the trophic guilds. And we also have the ordinal traits that, for example, the predator escape, it's like has a high potential of escape or low escape. Yeah, so we can classify as ordinal traits. So after you choose your traits, I hope I'm not going too fast. After you choose your traits, and you have wisely chosen them, then the second step is to choose the indexes that you are going to calculate functional diversity, because we have a lot of options here as well. So we have some indexes that don't consider the abundance or density of individuals. Yeah, and in some cases, abundance can be important. For example, if you are analyzing the presence of a new predator in that environment, and the predator is like extinguish some traits or some species, abundance can be important for you. So you need to think about that if you are going to use an index that don't consider abundance or that consider abundance. And some are strongly related to species richness as well. So it's like dependent on species richness, and we need to pay attention on that as well. This relation with species richness can lead the functional indices to redundancy as well. For example, FED, how entropy, and FD from Petsch and Gaston, they are dependent on species richness. So if you add a new species that is functionally identical to another that is already in your community, it causes an increase in functional diversity. But you are not having an increase in a new trait or a different characteristic. You're just adding a new species that is exactly the same. So it's kind of problem, a mathematical problem, yeah. So it considers that the distance between species pairs are identical, independent. So to correct these mathematical problems, these problems, in 2008 and 2010, two other measurements were proposed. One by Villager and another for La Liberté and Legend. And these ones, they are based on a functional space used by species in a multidimensional space. So this is why I was saying to you that we are going to transform our traits composition in distance, because it's plot in this multidimensional space, and it's calculated based on the volume occupied by species in this multidimensional space. So the first of them is the functional richness. Functional richness is the amount of functional space filled by the species and the traits. So if we have a great functional richness, it means that we have a great number of traits, and we have like a combination of unique traits. So for us to understand better, here in this figure, we have some black points in the graph. These points are the species, and if they are close to each other, it means that they share the traits, they have similar traits. And if they are far from each other, it means that they are very different regarding their traits composition. The size of the points is the abundance. So these species that are have a very unique traits, very different traits, they are in these points that are more extremes in the figure. And these extreme points, they are linked, and then the volume of this polygon is calculated. So if we have a great volume in this polygon, it means that we have a great functional richness. And if you have a small volume, it means that we have a low functional richness. So it means low functional richness, it means that we don't have a good combination of traits. We have similar traits in that community. In these indexes, so the rare species with extreme traits values, they greatly influence the indexes. So if you have just some species that are very different in your community, in your sample, they are going to be in these extremes, and they are going to influence the index. You need to think if this is a property that you desire in your question, in what you are investigating, because sometimes it can be like not something that will be important for your question, and then you maybe should remove these rare species. It depends what you are investigating. The second one is functional evenness. functional evenness quantifies the regularity in which the functional space is filled by the species and is weighted by their abundance. Yeah. So functional evenness is calculated based on the minimum spanning tree that it's similar to this figure that we have here. This minimum spanning tree, it arranges the species that are here again represented by these dots, these black dots. So it rearranges the species based on how similar they have their traits, the composition of traits, and also if they are close to each other, it means that they are more abundant. So species that are more abundant, they have these distances reduced, so they are going to be closer. So when we have low functional evenness, it means that we don't have a good distribution of abundance and traits in our community. It means that maybe we have some species that are more abundant, that have some traits that are more dominant in that community. So functional evenness is independent of other indexes, for example, taxonomical richness, functional richness, and functional divergence that I'm going to talk later. So it's also a good thing to investigate, because when Legendre and Villagers, La Liberté, Legendre and Villager, they propose these indexes, they say that we need to interpret the indexes together, because each one of them is looking for a part of the functional diversity, and we need to investigate them as a whole, like together, and interpret the responses of that community, not only to choose one or two indexes. So this is a good thing, because functional evenness is independent of the other, so you can interpret them together as independent. So functional evenness decreases when abundance is less evenly distributed among the species, or when functional distance between species are less regular. The third one is functional divergence. Functional divergence in a multivariated context, it relates to how abundance is also distributed between the volume of functional spaces occupied by species. So in this graph, again, each dot is a species. If the dot is larger, it's bigger, it means that the species is more abundant. If they are close to the center, because here this index, it calculated like a gravity center. If they are close to the center, we are going to observe a lower functional divergence. And it means that we have like a prevalence of traits and species. The more abundant species, it's like pulling the abundance to the center of this gravity that it's calculated based on this distance in the multidimensional space. So the functional divergence, when we have a high functional divergence, it means that we have a high degree of niche differentiation among species within our community, the community we are investigating. So the most abundant species are highly dissimilar, and that they compete weakly. So we can again think that if they are competing weakly and they are using better the resources, they have a more efficient resource use. So we also increase the ecosystem functions in that site. The other one is the functional dispersion. Functional dispersion is calculated in as a distance to the centroid. It's a modified measurement from the papers of Anderson from PermDisp. So functional dispersion, it's a mean of individual species to the centroid, of all species in the community. So if again, each dot here is a species, the dot is bigger, it means that the abundance of that species is also higher. And when we have these species that have more abundance, they are going to be closer to the centroid. This will reduce the distance to the centroid, and then the average, the mean of distances, and then we have a lower functional dispersion. Yeah? So when we have a lower functional dispersion, means that reflects that the overlaps of niches. When we have a high dispersion, it means that we have a differentiation of niche. Then again, the species are not competing, they are using the resources better, and we have an increase in ecosystem functions. So here, each species will be close to each other if they have similar traits, and far from each other if they have different combination of traits. Another index that is highly used as well is the CWM, that is the community weighted mean. I can say that it's like a summary of how the traits vary inside a sample or inside a community. So you can get all your traits, and then you are going to analyze each one of them separately. And you are going to see how that trait vary inside each sample that you have based on the abundance of species. Yeah. So the weighted average of a specific attribute in relation to the abundance of all species occurring in that locality. So let's imagine that we have a sample number 1 that is like my n here in this matrix. To calculate all the indexes, we are going to see that we need the matrix of species by samples or by the community that you are analyzing, if you're studying, so it's on so on. And we need a matrix with the traits by species. So let's imagine here we are analyzing our first sample, and we are going to see how the body size is in that sample. So we have the measurements, and the CWM gets the body size and do like a weighted average of that body size inside my sample based on the abundance of species that I have there. Yeah. So these are the indexes for alpha diversity, these ones that I showed you, the functional richness, functional evenness, functional divergence, functional dispersion, and the CWM. But as we can calculate functional diversity for alpha diversity, we can also calculate for beta diversity. And all the indexes that Dani showed you before, we can apply, instead of thinking of a taxonomic point of view, we can think about the functional point of view. So instead of analyzing the species, then we're going to analyze the changes in traits. So for all the measurements, all the indexes for beta diversity that we have on literature, we can apply for functional beta diversity as well. So we have the total variation between sites, that is the total beta, would be how my traits are different in my set of samples and environments. We also can partitionate functional beta diversity in the turnover. That is like how one trait is replaced by other, and the nestedness, that to see if my traits are nested. We also can apply the other approaches of, for example, richness difference. They analyze the difference of richness trait, the richness of traits between my types and the replacement as well. Same for the local contribution. We can analyze that site, how much it contributes regarding the traits composition to my entire beta diversity. So we can apply total LCBD. We could analyze the local contribution of traits to the replacement, the local contribution of traits to the richness difference as well. So those were my first two steps. Just remembering again, the first one is how to choose my traits that need to be wise, need to answer my questions. The number two is to choose the indexes that I'm going to apply to answer my question, my investigation. And the third step is computing the indexes that needs the more practical part. Anyone has questions? Can I move to air?
I don't see any questions right now. I think we can move.
Okay. So again, functional diversity is a very comprehensive topic, and I try to summarize the main ideas. But if you have any question at all, maybe you can, if you don't want to act now, you can send me emails, it's all good. So the idea here is to like give a simple talk, and it's more like a practical about functional diversity, but how to interpret the data. So here in the script that I have prepared, you are going to see that we are going to run, for example, some regressions with GLM and doing other analysis, but it's not my goal here to enter specifically on statistics and how to test, but to interpret the results that it's about functional diversity. Yeah? So everyone has this script, and this first part is to install the package we are going to use today. So this first part, we are going to use the FD mostly. So we can load here the package. It's okay? So we are going to first load some data set. We are going to use the dummy data set that is inside FD package. So we are just this first part is just to run the data set. We are going to define on that data set what are the data about species, and we are going to use abundance of species. We also going to need a matrix with traits. So just defining here, saying which thing is which. So we are going to have 10 samples here, 10 communities, and we have 8 species, and then we have the abundance of each one of them. For our traits, we have here a mix of traits. So we have numeric traits, we have factors, we have ordinary, we have binary traits as well. As you can see, we can run the analysis with missing traits. For example, for species 2, I don't have the information about the ordinary 1 trait. Yeah, so this trait here is just an example, but we can imagine, for example, that the numeric traits are body size and maybe the weight, body weight. We could think that The ordinary could be predation escape or something like that. The binary, the presence of spines, for example. Yeah. So for interpret it better, let's keep that in mind. That could be these examples that I said. So the first step is to run the distances, as we have a mix here of traits, different types of traits. We are going to run Gauer distance. We are going to transform our traits in a matrix of Gauer distance. If you have only numerical traits, you could use, for example, Euclidean distance. So this is our first second. So this is the metric that we are going to see, the distance between species regarding their traits. If they have like a similar composition of traits, they are going to have a low number. If they have a very different composition of traits, that the species are going to have a bigger distance. For example, these two, species 3 and 4, the distance is 0.82. So this distance are, they vary from 0 to 1. So 0.8 is like, they are very different regarding their traits composition. To run all indexes of alpha functional diversity, we are going to use this package, this function, sorry, that is inside FD package. The function is dbfd. We can call it here the help to take a look. So from this, we could calculate the functional richness, functional evenness, functional divergence, and functional dispersion, as well how quadratic entropy. And we could form groups of based on the composition, functional composition as well. We can calculate CWM. So all of them is inside the same function. Here we can see some arguments to run this function. As I said, we are going to need a matrix of species by size and another of traits by species. Yeah. We can weight the abundance if it is important for you for some species that are more important to the question you are raising. You can weight their abundance compared to the others. There are lots of other arguments here, but they are not like they are, if you want to investigate like deeply, but the main things is what I just showed you. If you want to create groups, for example, we can do a cluster, and then you choose the way that you want to do this cluster, and you're going to cut the tree to calculate functional groups, for example. This way that this function calculate functional groups, I think it's kind of, how can I say, it's only visual inspection, and you don't have a number or something like this or statistics to analyze if there is a significance change in that. You just make the tree, the cluster, and you cut whatever you want to make the groups. So it just visual inspection is highly recommended. So I think it's that from this. I will show you the output when we run the function. So let's go back here. So we are going to use here the three arguments, choose my traits, that is the distance that we create based on our distance. We are going to choose our matrix of species, and we are going to do a correction called Kelly's, something like that. This correction is made because when we are calculating this distance to plot in the multidimensional space, we calculate the distance between species based on the traits on Gower, but when we plot them in the multidimensional space, they are transformed to Euclidean distance. And Euclidean doesn't accept negative eigenvalues. So we need to transform, make this correction if our data has Euclidean distance. So it's just to correct. If your data is okay, the correction will not be applied, but we need to use this argument as well. So we can run here. It always gives this information here, for example, so, for example, species by species distance was not Euclidean, so the correction we asked was applied. If could not be calculated for some communities, as well for functional richness, right? And also for functional divergence. Why is that? Do you remember that I said on the first part of my talk that we need to have more species than traits in a community? If you have, for example, 4 traits, but you have the presence of only 2 species on that sample, you have it's not possible to calculate these indexes because you need to have more species than traits. So other things in here. So as I said, if our distance between traits and species is plotted in this multidimensional space, and it's usually plotted as PCOA. So this PCOA needs to choose the better explanation of the axis. So usually it's the first 2 of them. And it's saying here that the last 4 of 8 were removed. It's just for us to know how is the representativity of our data in that multidimensional space. So it also says the quality of this reduction. So it needs to be reduced, but how good is this representation? So this value here that is 0.65 is similar to R squared if we interpretate it. So for this one, it's a good representation. If it was lower than that, for example, 0.3 would not be a good representation. So we need to be careful when we are doing the interpretation of our indexes. And the last thing is that we enter the function with the distance created by our distance, and then it's not possible to calculate CWM. We need to enter the raw data to calculate CWM. But then we can do later here. So now we can call the output of our function. So the first one, means the taxonomic richness for each community. It's not our goal to look at. Yeah, how different species we have in each community. And then our functional richness for each community, we have, for example, 0.26. We have also community 8, that is 9 here, community 9, and that is 0.56. So it's like higher than the others. It means that this community has a more unique composition of traits than the community 1 or 2, for example. This is the quality, the functional space that I already told you before. The functional evenness of each community as well. So the first community has a great functional evenness. It means that the abundance in between the regularity in which the functional space is filled by the species and abundance is well distributed. Different, for example, the community 2 that has a lower functional evenness. Then functional divergence, also the numbers, functional dispersion, same as I told you, and the Howe entropy as well. It's okay here. Anyone has questions? Okay. So we can move forward. This next line here is for us to call if you want to see separately, for example, just functional richness, just evenness, just the quality. To calculate the CWM, we need to enter, as I said, the traits as raw data. So we are going to run again here the function with the traits as raw data. And then we call the answer. So the response of that, the output. Here, for example, for the numeric 1 trait, let's imagine that is body size. And then we have how that body size vary inside my community based on the abundance of species. So if I have, for example, some species that 3 species, let's imagine that we have 3 species that are very abundant and they have bigger body size. So the CWM will have a higher number as well because it's weighted by that species. That species have a larger body and they are more abundant. So the trait will be pulled by this information and will be higher. If I have species that are more abundant with smaller bodies, then this would have a lower number. Yeah, this is what CWM do for all traits. Okay, so how can we use this information? We can interpret functional diversity based only on that, what it means, functional richness, divergence, and so on. But we can use this information also to run other tests. For example, if I'm investigating how temperature changes my traits, how is the effect of my the temperature on my trait. So we can get this and run other analysis. Okay, so here we are going to gather our the output of our test as a data frame. We are going to create 2 predictors. One is a numeric. So for example, let's imagine we are investigating the effect of the percentage of forest in streams. So how important is on my functional richness, for example, this will be the first predictor. We can put here, for example, percentage of forest. My second predictor will be, for example, temperature, warming could be, huh? So we have like an experiment, let's imagine, and we have 3 temperatures. We have high temperature, we have moderate temperature, and low temperature. How this temperature is going to affect my functional diversity. Then we put everything together. We're going to run here a regression. Just here. To do so, first like, as I said, it's not my goal to go on to deep on the statistics, but just to see my distribution, to choose my family on GLM. On my correction, it's okay. We are going to do this function, beta hack, beta hack, then call the summary. So for example, we are going to, here we are investigating if the percentage of forest cover increases or decreases my functional richness. And we can see that yes, it has an effect, the percentage of forest in this data. So it's all fiction, but it's nice to interpret it. Let's do a plot here. Yep. So we could say that the increases on forest cover also increases functional richness. It means that when we have streams with more forested, that they're forested, we have a unique composition of functional traits. So we have here better use of resources, and we have more functions in this environment. This would be the interpretation. The second example is with the category as a predictor. So let's see here. I did a novum and it was not significant. Let's interpret these results here in the plot. So I use it as a response variable here, the functional divergence. So we could say that temperature has no effect, the increase in temperature has no effect on functional divergence, but all our treatments, we have a great functional divergence. So it means that for this community, this species that I'm analyzing, we have a great use of resources and a great functional divergence independent of the increase in temperature. This would be the interpretation of the data. Okay, guys. So these are some examples of how to run alpha functional diversity, how to interpret the output, the data, how to use to do other analysis. And then I'm going to show very quickly to you also about functional beta diversity. So for that, we are going to use these two packages. We are going to use this data set that I sent to you. I hope you guys have it. So we are going to use a data of species abundance and traits. And here we are going to standardize this abundance using the Heilinger. Also here, the first step is to transform our trait in distance. We are also having here, we can call it for you guys to see. We have like different types of traits. So we have feeding habit, this threat on or the species feed, the breeding, the potential of migratory, if they are migrant or not. So we have these more categorical traits, and for that we use also Gower distance to transform our traits. So we're going to run here how Gower distance, and then again we have distance between species based on the traits, the composition of traits. Then for beta functional diversity, we create a cluster using this function. So here we can see it, the species that are close to each other, they are similar in the tree. If they are regarding their traits, if they are far from each other, they have very different composition of traits. And we have here the height of the branches. So for functional beta diversity, we use this information, this distance between branches, to calculate how similar or dissimilar the communities are, how the composition of traits change between sites regarding this. So here we are going to use this function, beta function from Petsch. We are going to enter the species, the abundance of species in each site, the tree that we just created here with this function. And we are using the abundance. So here I say abundance is true. And we are going to use Jacquard dissimilarity. So as Dani explained to you, Jacquard, it does not give any importance for rare or common species. It's like equal. So this could be your goal, for example, depending on what you are asking. So we can run here and call the results. So we are going to have three. Using this function, we are going to have three distance metrics. The first one would be like the total variation. The second one would be the partitioning. So we can just separate these results with these functions here. First, the total functional beta. Then the richness difference, that this is the approach that I'm using in this example. And then the replacement of traits, we can call like the mean, the average of distance to know which contribute more, how is my total variation, because we have like this pairwise combinations, right? This distance between species, between sites in here, that is the better results. So for example, my total beta was 0.49. So it's kind of okay. It's not like a big dissimilarity in the composition of traits between sites, but it's not low as well. Keep in mind that it's also vary between 0 and 1. So it's like almost 50%. The average of richness difference and replacement. So we have that the replacement of traits contribute more to total beta than the richness difference. We can see like it's 34% of replacement, 14%, almost 15% of richness difference. Just a plot here to look at. This is the total, and then replacement, and then richness. As well, we can interpretate only that. Yeah. So for example, we could say that we have more replacement of traits than richness difference. I can have the same amount of traits between my sites, and we can think about the ecological interpretation of that. We could think that, for example, replacement can be related to dispersion, for example, between sites and other things. So we can interpret the ecological meaning of that. Usually we use this data to investigate what are the factors, the environmental factors that are shaping that results. So I'm going to run here example. This is the last part of my presentation. So we are going to open this data that it's about environmental data. We can call it here to take a look. So we have, for example, here, if you have in that site farms, if you have small buildings, high buildings, if you have industry, if it's fields, grassland, so on, other information. And we are going to see if any of these has an effect on my functional beta diversity. So for that, we are going to run RDA using this function. So let's call it here. I'm using only replacement here, functional replacement, okay? And I'm running with some part of the information of environmental data that I have, for example, small building, high building, industry, and fields. So we had a significant results. So we could say that these environmental factors are affecting the replacement of traits between my sites. Here, just for us to take a look. So we have, for example, here's industries that it's more, let's call it here. We have a low explanation, just the results of the RDA. Here we can call by each term to see which of these factors are indeed affecting. So we have like small buildings that is significant, and the presence of fields. So, and both have a positive relation with my replacement. So the presence of fields increases the replacement of traits between sites. This is the meaning. And only the first axis here on the RDA was significant. It's not a very nice plot, but it's not my goal here to do a beautiful one. It's just for us to take a look at the data, yeah? So I cannot see here, but small buildings is something like here, fields, that it's causing this variation in the replacement of traits. So this is what I have prepared. If anyone has questions, please let me know. It's just like a summary of what we can do with functional diversity. And I hope I will have increased your knowledge about this topic.
Thank you so much. I think that was very interesting too. So for me, it was really great to see the last example actually, because oftentimes we don't have continuous data and we just have yes, no, or, you know, categorical. And I think that's really interesting to see how this approach can be applied to that kind of data as well. And I like the goer's distance too for the mixed data. So I think these were both really interesting. Any questions for Francieli?
I have a quick question.
Okay.
Yeah, thank you so much for the really clear and comprehensive summary and walkthrough of the R script. This is really helpful. I was wondering about, you mentioned this in the beginning on your slides about if you have correlated traits and how you deal with those and how you choose which ones to keep in and which ones to remove. I've come across that in my work and just wondering how you approach that.
Yeah. Did you run any correlation like between them to see if they are significant? Yeah, and they are. Can you tell me which traits are correlated?
I can't remember in particular, but things like body size and then other sort of traits like respiration. Yeah, quantitative, both quantitative.
Yeah, so I think you need to think about your question. Which one of these two traits is more important for what you are investigating? Yeah, if it is just a respiration rates or if it's body size. Body size is much more complex trait. Okay, it is correlated to others, but it also has a bigger meaning because body size can be related to so many other ecological responses and effects biotic interactions. For example, I don't know which group you are talking about, but it could be, for example, a response to predation, to how they feed. They are related to body size. So maybe body size could be a best choice for you because it has like more meaning and it's like a wider interpretation of your data and the effects and responses. Yeah.
Thank you.
No problem.
Any other questions to either of the presenters, Danielle or Francieli? Well, if not, I'm sure we can email them with questions later. And I just wanted to extend my thanks to both of the presenters today for being here, for putting the presentation together and making yourself available. So thank you so much.
Thank you too. It was great being
Thanks,
here. Thank you.
Thank you again. The pleasure to be here. Thank you for the invitation.
Thank you. Thanks everyone else for